Alrighty. It's a real honor to be here and help recognize all that Sloan MoBE has accomplished and to contemplate the future. And I'm going to tackle now a concept that I think we kind of shattered yesterday for the indoor air environment, this prebiotic, probiotic concept. But let's pick up a few of those pieces and think about how that framework can apply to built environment water systems. And so, if we think about a glass of water, it's important to recognize there's a lot of microbes in there, about 10,000 cells per milliliter. And as Norm Pace is fond of saying, it's just a little extra protein. The question we're trying to answer is, where are these microbes coming from? And it's a very interesting, complex dynamic between, you've got your treated water coming out of the plant, and then it passes through miles of distribution system pipes, and then it arrives at the building. And there you have all sorts of interesting phenomena, such as high surface area to volume ratio, long storage time or stagnation when you're sleeping or at the office, reactive pipe materials, and nice warm temperatures that within the building envelope make occupants comfortable, but also make the microbes comfortable. And most of these microbes are completely harmless as far as we can tell, but there are definitely some bad actors that we worry about, and especially these opportunistic pathogens like Legionella and mycobacteria. And in the US and most developed countries, these are now the leading source of waterborne disease outbreak. And they're a real challenge because the aquatic environment is their native habitat. They are not fecal associated, and that's how our water safety and regulatory framework is set up for fecal pathogens. Now, they usually affect immunocompromised individuals, but they can affect healthy people as well. And it's important to also recognize there's a variety of exposure routes. Again, our regulations tend to focus on ingestion, but for example, Legionella and mycobacteria are thought to be spread via aerosols, and they cause lung diseases. And we live in a world of change, and there are a lot of trends right now that could be thought of as the perfect storm for opportunistic pathogens. So we do have aging infrastructure, as we heard about this morning in Flint, but also across the country. This is a crisis that we have to deal with, and we're going to have intrusion of microbes, but also nutrients that can support pathogens. We also have recycled water, so we're looking to new water resources. We have this push to lower the temperature on our water heaters, which reduces scalding and it saves energy, but it also puts that water within a range that's conducive to microbial growth. And finally, we've done some work on green buildings, which conserve water, but also lead to higher water ages and stagnation time, which is also conducive to microbial growth. And so our broad objective of our research is to work towards identifying effective engineering and management controls for water systems and drawing from fundamental knowledge of the microbial ecology and addressing issues like opportunistic pathogens, and we've done some work on antibiotic resistance as well, and to prevent the spread of disease and protect public health. And so, if you will, you can think of this as a prebiotic approach in that it's intentional control of the environment to select for the desired microbes, or a probiotic approach would be where you intentionally add the beneficial microbes. And so the question is, can this really work? And of course, they are complex systems, but there's reason to to follow up on this, and one is that water is wet. Water is prerequisite for life. It's a little bit different than the rest of the built environment, which is described as a desert, and a lot of the microbes we're detecting are kind of passing through. But as Jeff Siegel pointed out yesterday, that if we want to find the microbes, we look for that liquid water. Another argument is that we are already doing this. Engineers already manipulate temperatures and disinfectants and whatnot to control for pathogens, but it's been a bit of a black box blind approach. And then we're not trying to manipulate the human microbiome here, right? We're just trying to manipulate the water microbiome, and we know who the bad actors are. And there's some promising research out there. So for example, I was at a conference last week in Rome focused on Legionella, and there's work being done on identifying bacteria that are inhibitory to Legionella. And also some of the work by Nick Ashbolt is focusing on the amoebae, which play a very interesting role as hosts to a lot of opportunistic bacteria. And he's shown that if you can encourage them to eat other bacteria, then that breaks that cycle. And so all this is to recognize that the premise plumbing environment is a robust microbial ecosystem. You have all sorts of interesting things going on. You've got, this is a pipe cross-section, so slimy biofilm, here's your amoebae preying on the bacteria. It's an oligotrophic, low-nutrient environment, but there's high flow, and there's scavenging of nitrogen and organic carbon. You've got corrosion that produces hydrogen, and that's an electron donor for microbes. And even in a water heater, you've got a gradient of temperature, and you also have corrosion and hydrogen. And so the point is that, you know, in engineering and in the public in general, we tend to have this mindset of the only good bacteria are dead bacteria. And what we really need to think about is managing this microbial ecosystem. And there's some analogies, for example, with overuse of antibiotics. So, you know, we need to use them at times, but we need to optimize that use. And the same can be true for disinfectants like chlorine. And so through Sloan support, we were able to do some very fundamental research and look at this interrelationship between the water treatment plant and that water provided and what's going on in the premise plumbing. So we had these standardized rigs, one installed very close to the treatment plant, one further out in the distribution system. And we saw a very interesting effect that even though we tested different pipe materials and stagnation, that things really clustered by which water utility they were installed at. And this example of utility A and B, they actually draw from the same water source, just maybe a few hundred meters away, but it's different treatments at the treatment plant, so that matters as well. And so this means that the municipal water quality is an overarching factor shaping the microbiome. And when we look at canonical correspondence analysis, we see that it's things like disinfectants and nutrients and pH that are shaping the microbiome. And there's no better proof of the hypothesis of the importance of the municipal water quality than what unfortunately happened in Flint, that switching to that corrosive water supply, as Mark talked about, led to a Legionnaire's disease outbreak that sickened 92 people and 12 people died. So as we're contemplating other shifts in water supply, a big one is recycled water, and it makes a lot of sense to take pressure off of water resources by recovering treated wastewater and treating it to a very high standard, Rita Caldwell talked a little bit about this yesterday in the panel. And the question is, even if you treat it to the highest of standards, the chemistry still is different. And so we need to be proactive and think about what those effects might be, both on opportunistic pathogens, antibiotic resistance, and we're even moving towards not just irrigation, but direct potable reuse in parts of the country. But again, it's not just ingestion that's the relevant exposure route. And so we're just completing this survey where we went out in the field and sampled recycled water distribution systems in 4 communities, and then compared with the potable system as a baseline. And so we sampled the raw water before treatment, the freshly treated water at the point of entry to the distribution system, and 5 points of use. And no surprise, the recycled water had a lot more nutrients. I'm showing the organic carbon here, but also nitrogen and phosphorus were higher. And using qPCR to monitor total bacteria and opportunistic pathogens, it's no surprise. So reclaimed on the right, we did have more detection of bacteria. Now in the potable water, so the X's show that freshly treated water and the bars are at the point of use. So we did see a little bit of regrowth of Legionella and mycobacterial numbers. And in the reclaimed water, we actually saw some reductions of Acanthamoeba and Pseudomonas aeruginosa. But we saw measurable regrowth of Legionella in these systems. And it's interesting because Pseudomonas has been identified to produce some inhibitory substances against Legionella, so that's interesting. And we also saw regrowth of mycobacteria and Naegleria fowleri, so that's also known as the brain-eating amoeba. So another thing to scare folks, or just see if you're paying attention here. Now, fortunately, the exposure route for this, you have to actually snort the water up your nose, and so it's a very rare infection. Looking at the microbiome, we see that there is a unique signature of the reclaimed water compared to the potable waters. And then we've been doing some metagenomic sequencing and not having too much trouble in finding markers of many of the opportunistic pathogens that we're interested in. So that's thinking about distribution systems, shifting water chemistries. So getting back into the premise plumbing, there's a lot of things you can manipulate. These are some of the things we've looked at. I'll focus a bit on temperature and pipe material. So this is a study where we used these glass bottles to simulate water heaters and added PEX, which is a plastic pipe material, and we know it leaches organic carbon, and copper, which is known to be an antimicrobial. And so we increased the temperature with time, and we saw a reduction in bacteria, as well as Legionella. The colored ones are the ones where the temperature was reduced. Where the temperature stayed stable, the bacteria preferred, or the Legionella preferred PEX in this study. Interestingly, the vermamoeba, which is a host for Legionella, was the opposite. It preferred copper. And we've done some work scaling this up to a pipe rig scale, and it's no insignificant feat to operate one of these. And I've presented some results previously on how when the temperature is at 51, there's this sweet spot where it actually selects for Legionella at the tap when there's low flow. So it's not quite hot enough to kill it, just enough to select for it. This is some newer data where we've looked at the composition of the microbial communities, and interestingly, there's that same threshold in the biofilm where we see a shift in microbial community. And not to forget the mycobacteria. I've had the pleasure to work with Joe Falkenham, And this study actually originated from him reading the supplemental information of Norm Pace's shower head paper. And he noticed that when mycobacteria were absent, methylobacteria were present, and vice versa. And so we just did a lab demonstration and published this. So these are consortia of methylobacteria, and when there's a biofilm of that, the mycobacteria do not establish. But they love the normal tap water biofilm. And so can it work? Again, I would make the case that we are doing it, but I think now we have the opportunity to look inside the black box and understand why. Prebiotic is more straightforward, right? Adding disinfectants, manipulating temperatures, pipe materials. Probiotic is maybe further out there in terms of intentionally adding microbes to water, but an example where we're doing that already is with these biofilters. And I just also wanted to mention again the opportunity that Sloan brings to collaborate with other scientists and brings you with people that otherwise you wouldn't have met or interacted with, and sparks new collaborations. And so I had the opportunity to work with Ruth Berkelman and Gary Russell and Allison Chamberlain and co-organize this workshop on Legionella, and it was the first interdisciplinary workshop on Legionella in 25 years. So thanks to Sloan for that. And we did recently publish an article if you want to read about that. And finally, I just encourage us not only to learn from different disciplines, but different countries, as we've been doing this morning. Much of Europe, for example, they don't chlorinate their drinking water, and they seem to be doing okay. So I think the funding sponsors, as well as my collaborators and students, and hope we might have a time for a question or two. Thank you.
We have time for a couple of questions.
Thank you for a nice talk. What is the effect of chlorinating the water on microbes in the pipes?
What is the effect of chlorinating? I didn't show the data here, but we've run head-to-head experiments with and without chlorine, and it has a very strong selective effect. You get a very different microbial community. And in terms of direct effects, it's a strong oxidant. And so you select for microbes that can survive that kind of oxidative attack. So that's part of why it's hypothesized that mycobacteria survive so well in chlorinated drinking water. They have a very waxy protective coating. We are interested in whether it selects for antibiotic-resistant microbes. We've seen it in other studies, and we have some preliminary data where we're looking at antibiotic resistance genes, but not quite ready to publish that yet.
So I'll ask one question, and that's related to the outcome of all of this is, in terms of exposure, do we have a good idea of, when you integrate across all the water uses in a home, how much of our exposure to the pathogens in our home water come from consuming it versus aerosolization and healing?
We really need to do that work, because even, I showed the example of inhaling aerosols in a shower, and we know that cooling towers, those aerosols are a source of Legionnaires. But there's evidence too that, when you swallow the water, sometimes it goes down the wrong pipe. That's called aspiration. And so it would be very helpful to have a sense first, what are the loadings from different sources, and to really know who's getting sick from where. Yeah.
Right, there's no more questions. Lunch awaits us. Thank you again, Amy.